Oggi parliamo di nuovi trattamenti per il mieloma multiplo ricaduto refrattario con la professoressa María Victoria Mateos, che abbiamo incontrato qui a Roma, dove è in corso il convegno Current Challenges in Hematology, giunto alla sua seconda edizione. Professoressa, let's talk about anti-BCMA bispecific antibodies. Which are the most promising ones and which results do we have from the clinical trials?
Thank you very much, Anne. From the BCMA bispecific monoclonal antibodies point of view, we have a list of at least 5 BCMA bispecific monoclonal antibodies under development for the treatment of relapsed and refractory multiple myeloma patients, majority of them exposed to the 3 conventional drugs, proteasome inhibitors, immunomodulatory drugs, and anti-CD38 monoclonal antibodies. There are 2 BCMA bispecific monoclonal antibodies so far approved, teclistamab and elranatamab. Both had been evaluated in triple-class refractory myeloma patients, and the overall response rate is of approximately 60%. Nearly 1/3 of the patients achieved complete response, and what is important, the durability of the response. The durability of the response is longer than 1 year, and the median progression-free survival is between 1 year and 1.5 years, indicating that all these BCMA bispecific monoclonal antibodies cover the unmet medical need we had for the triple-class refractory myeloma patients.
And are there other bispecific antibodies in development directed against other targets than BCMA?
Yes, sure. And beyond BCMA, it is possible to target GPRC5D or even FCRH5, and we have talquetamab and forintamab targeting GPRC5D. Talquetamab is already approved, again, in the triple-class exposed and refractory myeloma population, and the efficacy has demonstrated an overall response rate of approximately 70%, with median progression-free survival of approximately 14 months. Similar data with forintamab, although the clinical development plan is a bit late, and this means that the follow-up is shorter. But what is important is these GPRC5D bispecific monoclonal antibodies had been evaluated in patients previously exposed to BCMA-targeted therapy, and they work. Anthebostamab has been evaluated in relapsed refractory myeloma patients, and the efficacy and safety results are pending, and they will be released in the upcoming months.
During your presentation, you talked about the possibility in the future to use combinations of different bispecific antibodies. Which are these combinations and which are the data so far?
Yeah, this is the next step, and BCMA as well as GPRC5D bispecific monoclonal antibodies combine quite well with either anti-CD38 monoclonal antibodies or immunomodulatory drugs. There are some bispecific monoclonal antibodies in combination with proteasome inhibitors, but from my point of view, we've seen in preliminary phase 1-2 clinical studies very encouraging efficacy data about the combination of bispecifics with daratumumab, with IMiDs, and I personally consider that the role of combining BCMA or GPRC5D with either lenalidomide or pomalidomide is quite attractive, but why not to move forward combining both bispecific monoclonal antibodies targeting GPRC5D and BCMA? And we've seen some data about the combination of teclistamab plus talquetamab, and there is a specific population of myeloma patients with extramedullary disease that they responded very well to this combination. Extramedullary disease, pure extramedullary disease, is actually one of the high-risk features we see in patients with multiple myeloma. To have the possibility of combining these 2 bispecific monoclonal antibodies and to offer to the patients responses, deepened responses and durable responses, I think that it is very attractive.
And let's talk about cellular immunotherapies, that is CAR T-cell therapy. Which CAR T do we have now, and in which patients with multiple myeloma can we use them, and which are the results?
Yeah, right now we have two BCMA CAR-Ts approved for, again, triple class exposed and refractory myeloma patients. One is ID cell and the other one is cilta-cel. Both covered the unmet medical need. ID cell resulted in an overall response rate between 70 and 80%, with a median progression-free survival of 9 months, median overall survival of 2 years, with an excellent toxicity profile. Cilta-cel is another BCMA CAR-T. I would say that more effective because more or less in the same population, the median progression-free survival reported has been of approximately 3 years. What it is something unprecedented for myeloma patients that had been exposed to everything and they had received a median of 6 prior lines of therapy. These are the two BCMA CAR-T so far approved, but the next step is to move with them to earlier lines of therapy. And we have 2 phase 3 clinical studies with these two CAR-Ts. ID-cell has been evaluated in the KarMMa-3 in patients after 2 to 4 prior lines of therapy, and we are pending of the final approval by the authorities. ID-cell showed to be better than whatever standard of care, but it's true that there is not any significant benefit in overall survival, although it's true that crossover was allowed, and this can explain the lack of benefit in overall survival. And with the cilta-cel, we have a phase 3 randomized trial conducted in patients after just 1 prior line of therapy, and the progression-free survival for ID-cell is significantly longer in comparison with the standard of care. So we can envision that maybe these two BCMA CAR-Ts or just cilta-cel, but hopefully ID-cell and cilta-cel will be approved in earlier lines of therapy.
And finally, looking towards the future, are there other interesting and promising CAR-T cells in development?
Yes, sure. There are multiple possibilities in order to improve the efficacy, in order to improve the safety profile. But for example, the academic BCMA CAR-Ts are an excellent opportunity in order to increase the availability for the use of CAR-Ts across the different countries. And in Spain, we have an academic CAR-T with very promising efficacy data. But there are other CAR-Ts, and for example, anito-cel developed by Kite is an interesting BCMA CAR-T. It has a small size, it can penetrate very well in the extramedullary disease, and the efficacy and safety data recently reported are amazing because the overall response rate and the complete response rate in more than 80% of the patients has been sustained across the different subgroup of patients. So we need to wait longer follow-up in order to see how all these new approaches will be placed in the treatment landscape for patients with myeloma.